All right, so now we need to talk about how pathogens evade immune response. And what I just showed you in here, well, it seems very uh, basically efficient way of protection against pathogens. We have many different layers, and actually we have more mechanisms as well. So this schematic shows only the uh, crucial parts or the major parts of the immune defense. We have more stuff, but it would have made this schematic too complicated. So as I mentioned, we have the so-called evolutionary arms race, and as a part of that arms race, we have evasion mechanisms. Evasion mechanisms. So you're a pathogen, you're dealing with a complex immune system with a lot of different defense mechanisms. So you have to, if you want to survive, you have to find ways to evade. So evasion mechanisms could be categorized uh, to two groups, two major groups. Could be, it's a rough categorization. Some evasion mechanisms, the pathogen changes something in itself in order to evade. Some other evasion mechanisms, it changes things in the immune system in order to evade. And sometimes they use both types of mechanisms. So either I have to change something in myself, maybe change some of my antigens or some of the molecules that I produce in order to trick the immune system into believing that I'm okay. Or sometimes I need to remove molecules and stuff that are produced by immune system. All right. So how about how about mechanisms for pathogens which are intravesicular? Intravesicular. What can they do? I'm starting from changes in the immune system. We have some pathogens like Mycobacterium tuberculosis or this one Legionella pneumophila, which inhibit phagolysosome formation. That's the way that they work. The CGD is an immunodeficiency in which the production of ROS. Yeah, in a sense. But CGD is a genetic disease. Right. So there are parallels. There are parallels between the two. But the way that they work is not by inhibiting the production of ROS. Exactly. But this is not that. This is not that. Mycobacterium tuberculosis inhibits the formation of phagolysosome. It doesn't let the lysosome bind to the phagosome. Why? Because, well, if it binds, Mycobacterium can still resist the situation. It has evolved ways, developed ways to protect itself. But the point is that the phagolysosome formation is necessary for cutting some of the uh, protein antigens into peptides, and those peptides will get loaded at the surface of MHC, and then the T cell activation and the rest of the things would happen. So if you can block the phagolysosome formation, you have sort of blocked the first step in antigen presentation, or you have downregulated antigen presentation, and it helps in the rest of the process. Some other pathogens, like Mycobacterium leprae, which is the infectious agent that causes leprosy, has developed ways to inactivate reactive oxygen species. And we also have some other bacteria that produce catalase. Catalase degrades hydrogen peroxide. Exactly. So staphylococci are catalase positive, so they can inactivate hydrogen peroxide. And some other pathogens have come up with a more ingenious way. So they live inside phagosome as long as the situation is okay. But when things are getting worse, they break it and jump into the cytoplasm. So they jump out of the phagolysosome and then they continue the rest of the life cycle inside the cytoplasm. So they are intraphagosomal at first, but then they become intracytosolic. So listeria has this ability, and the enzyme which it uses for this process called hemolysin, because it can also lyse red blood cells. Probably the most interesting type of evasion mechanisms, however, have been generated or developed by viruses. So I told you about one of them. So here we have a virus which has infected a cell, and the cell is showing its antigen at the surface of MHC class 1 to CD8 T cells. So some viruses, like adenoviruses, have learned to decrease the production of MHC class 1. Exactly, because they know that if cells have sufficient number of MHC class 1 molecules, they will show their peptides, CD8 T cells will come and they will lyse the host cell. So they have developed ways to downregulate MHC class 1. In that way, they can get rid of the CD8 T cells. But we know that if you downregulate it too much, NK cells would come. So they have not evolved a way to decrease the activity of NK cells. So in that sense, they are not very strong in evading our immune response, blocking MHC class 1 expression. However, another group of pathogens, pox viruses, have developed a better way to do the process. Exactly. They know that if they downregulate MHC class 1, then NK cells would come. So they have thought a better way. Well, of course, there hasn't been any thinking process. This is an evolutionary process. But if I want to sort of make an analogy with the way that human beings solve problems, I would say they've come up with a better way by thinking this through. So instead of downregulating MHC class 1, they decrease the loading of MHC class 1. They preclude MHC class 1 molecules from binding their peptides so that the MHC is still present at the cell surface, but it lacks sufficient number of peptides. However, we know that if CD8 T cells detect even a tiny bit of peptide, they start producing interferon gamma. What does interferon gamma do? It increases the number of MHC molecules. And if you have a high number of MHC molecules, chances are that some of the peptides would stick to their surface and will move to the basically surface of the cell and activate CD8 T cells. For that reason, they have developed a second evasion mechanism. They produce decoy receptors for interferon gamma. Decoy means a fake receptor which can be produced. And these decoy receptors are produced by the virus. And unlike regular receptors, which are present at the cell surface, these receptors are secreted into the environment. So they will go and neutralize, they will go and bind interferon gamma molecules produced by CD8 T cells, and they preclude the interferon gamma from reaching the target cell and inducing the production of MHC class 1. So they have two evasion mechanisms. They block the loading of the peptide at the surface of MHC. That's the first one. The second one, they block interferon gamma molecules from reaching their target. And in this way, they can basically stay away from T cells as well as the NK cells. So that's a very good evasion mechanism. Sure. If MHC molecules, even if both MHC class 1 and class 2, when they are present at the surface of the cell, there are some self-peptides at their surface. So it's basically MHC molecules are never empty. So they always carry something. So if that something is from a pathogen, they will present the pathogenic peptide. It would be self-antigens. And no. It's no, because to sort of the cleft of the MSC has to have something, and it usually has a peptide. So if a cell is a normal cell, all of the peptides at the surface of MSC are self-peptides, to which CD8 or CD4 T cells do not respond because they have developed tolerance to those self-peptides. But if there is an infection, then the peptides derived from the pathogen will be loaded either at the surface of MSC class 2 if it's intraphagosomal, or MSC class 1 if it's intracytoplasmic. But perhaps the most fascinating mechanism of immune evasion is by cytomegalovirus, by CMVs. CMVs are DNA viruses. These are relatively big viruses, and they have sufficient space in their genome to carry different genes. I told you about arms race. So in the process of arms race, it's not always trying to make a new weapon. Sometimes in the process of arms race, you have basically espionage. Okay, and espionage, you try to gather information, including the so-called industrial espionage, especially if that industry is military industry. So you go and steal information from your opponent or rival country about the way that they make weapons, about the way that they make weapons. It's part of an arms race process. It's part of an arms race process. All right. So CMVs during their evolution have stolen something from us. Some people call it gene hijacking. Gene hijacking. They've stolen the information about how to make an MHC molecule from us. Well, we don't know it for sure. It's a hypothesis, but it's a hypothesis that is very likely to be true. How do they do that? Well, they enter the cell, they block the expression of native MHC molecules. They remove it from the cell surface so that CD8 T cells cannot lyse the target cell. So that's the first part of the story. But what should we do with NK cells? Because NK cells are always looking for the presence of MHC class 1 molecules. What could we do? They have stolen the information about the structure, about they have stolen the genetic code of the MHC1 molecule from us. So they've internalized it. They've changed the code so that they can make a molecule which looks like MHC, but it doesn't perform any function. All right, it's a fake MHC molecule. It's a fake MHC molecule. Where have they obtained it? Very likely by stealing our own genes. It's an example of espionage. It's an example of stealing militarily important information. So they put that fake MHC in here, and by that way, they get rid of CD8 T cells because there is no native functioning MHC molecule, but they also trick NK cells into believing that this cell is okay. So NK cells wouldn't do anything to lyse the target cell. And that is the way that CMVs work. It's a very ingenious way of doing the thing. So how about immune evasion? So these were for viruses and intracellular pathogens, the ones that I told you in the beginning. Phagolysosome formation, they inhibited, inactivate reactive oxygen species or jump into the cytoplasm, as well as these different mechanisms to evade CD8 and NK cell response. How about extracellular pathogens? Well, we have different mechanisms like resistance to phagocytosis, inactivation of complement, and stuff like that that I have not written in here, but you can find it in the textbook. We have the second group of evasion mechanisms as well, the ones in which the microbe changes or the microbe alters its structure in a way to evade the pathogen. So we have these four different sets of mechanisms. Change in the microbes, especially microbial antigens which are targeted by the pathogen. So one way to evade is for me as a species of a microbe to have multiple antigenic variants. Multiple, not it's not isotope, multiple epitopes. Epitopes. Isotope is the radioactive stuff. So pneumococci are a good example of that. Pneumococci can cause lung infection as well as the CNS infection. But the point is that when you say Streptococcus pneumonia, and actually you can see it in rhinoviruses, the ones that cause common cold, they have many, many different types, and each type has its own antigen. So as a species, they have different subtypes. And when you have a disease with one of them, when you get infected with one of them, antibodies are produced. But those antibodies are not going to be able to bind to other subtypes of the same species. So you get the disease with the species number one or subspecies number one or a strain number one. So you develop antibodies against it, but those antibodies are not efficient against the rest of the subspecies or the strain. So that's the way that they work. Or it's the same for rhinoviruses. You have almost 100 different types of rhinoviruses. I can get common cold with rhinovirus, I don't know, type number 23 today, develop some antibodies, but then 2 weeks after this, I get another common cold with another rhinovirus of another subtype. So that's one way that they evade. They evade it as a species. The second way is to mutate the antigens. So mutations happen in a lot of viruses. A good example is HIV. So HIV is a retrovirus. It has an RNA virus. It has an RNA. It has an RNA, and the target at the surface is this antigen, glycoprotein 120. So antibodies are produced against it, and they can neutralize the virus. However, when the virus infects a cell, RNA will be internalized. RNA is reverse transcribed to the so-called complementary DNA, and this complementary DNA will get integrated into the genome. and then it gets transcribed and produces proteins for the new virus. That is why it's called a retrovirus. So this target cell could be a T cell, a CD4 T cell, or it could be a monocytoid cell like a macrophage. The point though is that this reverse transcriptase makes a lot of mistakes. Making mistake is usually not good. But if you want to mutate something, making mistakes is very good for you. So this reverse transcriptase makes a lot of mistakes. It's called an error prone. It's called an error prone enzyme. What is an error prone enzyme? An enzyme which makes a lot of mistakes. So those mistakes would lead to a lot of changes in the cDNA, and it will lead to a lot of changes in the proteins which are produced in here. So this is the original virus. The antibodies can target its surface antigen, but the viruses which are produced have different glycoprotein 120s, and antibodies cannot bind to them anymore. So this is an example of mutations, usually tiny mutations. These are usually point mutations, one nucleotide changes at a time, that protects the pathogen from the immune response. These are different GP120s, and it keeps going on and on and on. So every time that the cell is infected, new GP120s are produced, and our antibodies cannot catch up with them. So that's one thing. This process, people call it point mutation. Some people also call it drift. Drift. Drift means a small change. Drift means a small change, okay? Shift is entirely different. Shift is big change. An antigenic shift does not happen everywhere. Only a particular group of viruses do that. Influenza viruses are able to make major changes in their antigens. Why do they do that? Because they have a genome which has multiple parts. 1, 2, 3, 4, 5, 6, 7, 8 RNA segments are present in the genome. They are separate from each other. And what happens is that influenza viruses have a lot of different hosts like human influenza virus, avian influenza virus, porcine influenza virus, which infects pigs. Different organisms have their different influenza virus. But sometimes two influenza viruses can infect the same host. Like a bird, a chicken can get infected by human influenza virus as well as the bird influenza virus. What happens is very bad. In the body of that chicken, a chimeric virus is generated. Part of the genome comes from human, part of the genome comes from the bird influenza virus. And this generates a new virus which basically is the population is completely unprotected against it. Because the human population, for instance, has had exposure to human viruses or maybe the avian virus, but this is entirely new virus. This is called antigenic shift. Another group of pathogens use genetic recombination. It's different from point mutations. So the genetic recombination happens in trypanosomes. These are pathogens which can cause sleep sickness, for instance, trypanosomiasis, different diseases, and another disease named Chagas disease. So these trypanosomes have an Achilles heel. Achilles heel is a metaphor for a point of sensitivity. Okay, you're a very strong person, but one area of your body is very sensitive to attacks, and that is the story of Achilles. Okay, so everywhere in the body was kind of very strong, resistant to arrows, but the heel was very sensitive due to the story of his mother holding him by his heel when he was sort of immersing him in that water and stuff like that. So these guys have an Achilles heel. And that Achilles heel is a glycoprotein at their surface, which is targeted by the antibody. The point, though, is that they can change this target all the time to DNA recombinations. They can change it all the time. And whenever an antibody is produced against the protein or the glycoprotein at the surface, they undergo a DNA recombination, they change that protein into a new type of protein, which is not targeted by the previously generated antibodies. So new antibodies have to be generated. And then they can keep this tens of times, I think 100 times, they can change their surface glycoprotein. This is not mutation in the old sense of the term or the point mutation. This is genetic recombination that happens at the DNA level. And finally, we have some pathogens like Plasmodium, which have antigenic variation as a part of their life cycle. It's not kind of mutation. They change all the time. They are kind of shapeshifters. They change their shape and antigens all the time. So in the beginning, the mosquito bites. This is the particular type of the pathogen that enters the body. It has a morphology as well as its antigens. So body starts producing antibodies against it. Then they go inside liver, they change their antigens. Then they come out, they change their antigens again. Then they become gametocytes, they change their antigens again. And during this process, immune system cannot catch up with them because they're always changing their shape and they change their antigens. So that's their way of evading immune response. So having multiple antigenic variants, point mutations and shifts, somatic recombinations, and basically changes of antigens during life cycle. So this is the end of the story and the summary in here. Pathogen type and localization are very important in the process. Activation of macrophages, both TL1 and NK cells, is an important defense mechanism against intravesicular pathogens. Interferons are important players in defense against viruses, CD8 T cells and NK cells, lyse virus infected cells. We have antibodies for defense against pathogens in the extracellular space, and we have a lot of ways for the evasion. Okay, so this is the textbook, and this is the quiz. Write down the answer to this quiz I gave you. Okay, so take it back. This is yours, I guess. This is yours. Okay, so this is the multiple choice question that you need to answer. And you don't need to give it back to me. Keep it for yourself because those questions will be, you will see similar questions in your exam. All right, so which of the following is not considered a mechanism of immune response against intracytosolic viruses? A, interferon response, B, NK mediated cell lysis, C, CD8 mediated cell lysis, D, activation of macrophages by NK cells. Is not a mechanism of defense against intracytosolic viruses. Do we have interferon response against viruses? Of course, we have interferon response against viruses. Can NK cells lyse a virus infected cell? Of course, NK cells can lyse a virus infected cell. When the MHC goes down, NK cells lyse. Can CD8 cells lyse the infected cells? Yes, they can lyse the infected cell. Do macrophages get activated by NK cells? Yes, they get activated, but it's not part of the classical defense against viruses. It's more important for intravesicular pathogens like mycobacteria. All right, end of the class. So I expect you guys to be able to draw this thing and memorize the interactions. Well, understanding is more important than memorization, mere memorization, but some of these things you need to memorize. And if you memorize this well and the interactions well, then it will give you a good, basically, the basic knowledge so that you can build up the rest of the knowledge that you require on top of it. Okay, end of the class. Thank you.